Hello, everyone, and welcome. Today we'll be talking about secondary lymphoid organs, namely the lymph nodes. I hope you've all been well. I know it's been a while since I made a video, but it occurred to me the other day, it's time to finish this series. I'll be doing the remainder. It's 10 parts for general immunology, but then there's like subparts sometimes, like this one is going to be 2 parts. And then afterwards, I hope to go into like more specialized topics in immunology, like transfusion reactions, hypersensitivity, blood typing, all these things. And also, I'd like to get into some microbiology. So that'll be fun. But we're doing our best, or we're trying to, right? We're balancing. So let's orient ourselves. This lecture is going to be a little bit unusual because up to this point, all we've been talking about is the what, like what is happening, what is the B cell doing, what is the T cell doing. Today we're going to talk about the where. So where are the B cells and T cells doing the doing, right? So in several regards, today is going to be a microanatomy lecture, which is just a pretentious way of saying histology, but that's a pretentious term too. It's just a mini geography lesson today, guys. Um, So to do so, we should probably just orient ourselves in seeing how lymphoid tissue is categorized. It can be categorized as primary or secondary, and you have bone marrow, which is the mother of the primary, and that's the origin of lymphocytes. By and large, and pretty much any blood cell is going to start its story off in the bone marrow. And you have the thymus, which is that magical academy that makes sure that your T cells don't destroy you. That's also considered a primary lymphoid tissue, right? Then you have your secondary lymphoid tissue. This is majority of your lymphoid tissue actually, and that's going to include your and I say majority, not necessarily in a quantitative sense, but more so in like how diffuse it is. Um, that's going to be your lymph nodes, your spleen, and your mucosal associated lymphoid tissue known as your MALT, and all the pretentious terms associated with it, like galt, salt, and balt. Um, but malt is malt, right? Um, so this is just the basic schema. Today we're going to be talking about the secondary lymphoid tissues, and we're going to start with the lymph nodes because really, if you can understand a lymph node, it makes understanding everything else easier, including your spleen, your blood filter, and the malts. Also, your tonsils, and the adenoids, which are just like your upper tonsils, are lymphoid tissue as well. So there's that too, and some people, it's not really set in stone yet, but when we talk about the appendix, a lot of people are like, "Oh, it exists just to give general surgeons a headache because it has to get removed in appendicitis." But some are like, "Oh, it's a microbiota reservoir, our gut bacteria reservoir," and some are like, "Oh, it has some immune system related to associated to the galt." Um, so I'm not gonna put appendix in here because appendix is still controversial at this point in time, and we'll document that for 2022. So, what is a lymphoid follicle? You're going to hear this term in germinal center. They're almost sometimes used interchangeably. They're not necessarily always the same, but more or less. So, a lymphoid follicle is going to be a follicular dendritic cell surrounded by B cells. Okay. Um, Follicular dendritic cell has very little to do with dendritic cell. So just because it has dendritic cell in the name, you think, oh, maybe they're related. Unfortunately, it's one of those non-intuitive things, as you can tell medicine and biomedical sciences are full of. The two are not related. So try to keep them distinct in your head. And also, I would recommend for you that you develop a schema, and the schema will involve having um, categorization of dendritic cells with T cells and follicular dendritic cells with B cells. So please try to keep those two straight with each other. So what is a follicular dendritic cell? It's not a dendritic cell. Oh, we know that much at this point, right? I want you to regard the follicular dendritic cell as this big sticky thing. Or you can think of it as a library. Not to go on a tangent, but When we talk about like great libraries, you can't help but think of the Great Library of Baghdad, which, if you don't know what it is, in the Abbasid Caliphate during the medieval era, it was a very remarkable type academic institution and library that really drew on the academic traditions of the Byzantines, which was a Greco-Roman tradition, and the Persians, which was a Persian tradition from what is modern day Iran, part of Iraq, Kuwait, and Central Asia. But also it drew a lot from the ancient Indian tradition as well over in Persia. So when you had the Great Library of Baghdad, Bayt al-Hikmah, we call it in Arabic, House of Knowledge or House of Wisdom, um, it really inherited these two academic heritages and really made the best of both worlds. And it was a great point of advancement, both in the scientific and medical sense, but also with regards to theology and philosophy and botany and alchemy, which would later give on rise to chemistry and literature and everything of this nature. So, and even like, Canon of Medicine by Avicenna, that's just the Latinized name. I'd rather say Ibn Sina, his Arabic name. He was really born out of that general atmosphere of that Abbasid academic tradition. Sorry, history is on my mind lately. But for all intents and purposes, you can consider the follicular dendritic cell as a great library of Baghdad, because really that's what it is. It is a sequestration point or great gallery of antigen. And why is it that way? Is it because it's really sticky because the little maintenance people in our bodies painted them with glue and they became really sticky? That would be cool, but no, they're actually sticky on the basis that they're very rich in opsonin receptors. And so you remember when we talked about opsonins before, an example I thought was quite funny I like to give is when we talk about, say, antibody acting as an opsonin, the FAB regions will bind an antigen, and the FC region will bind the FC receptor on a macrophage, and the macrophage will grab that FC region via its receptor and drag it in and destroy it. But because the FC region is bound to the FAB region, and the FAB region is bound to an antigen, it's kind of like if I'm going down, I'm taking you down with me type thing. Hence, the macrophage will also take in the antigen and destroy that as well, right? And that's, in that case, the antibody acted as an opsonin. That's just one example, but basically here the follicular dendritic cell can grab opsonized antigens quite well and just hold them up. And this will be super important because by being a big library of antigen, it attracts a ton of scholars, and these scholars are B cells looking to advance their academic careers, right? And that's really a schema I want you to keep in mind when we talk about the lymphoid follicles inside the lymph node and other lymphoid tissue. It's just basically an aggregate of B cells looking to advance themselves, and they can't reach their full maturation without finding their cognate antigen. Where might they find their cognate antigen? At the library where antigens are stored. And in the case of our body, follicular dendritic cell is a big part of that for the B cells. So, what happens in a germinal center? And I have this little star in a ball for you. Imagine this whole thing as your lymphoid follicle, right? This circle. The central region, we'll call that the light zone, and the green is a lighter shade of green there for that reason. That's the light zone where the follicular dendritic cell resides. And why is it called the light zone? Is it because the little maintenance men in our bodies painted it that way? No. It's just that the concentration of B cells in the light zone is a little bit less as compared to the other part, which is the dark zone, which is the darker part of the circle. So that's what it is. Just have the schema. We have a germinal center, lymphoid follicle, in this case, lymphoid follicle, light zone, dark zone. Inside the light zone, we have inside the light zone, we have the follicular dendritic cell, and it's going to be swarmed by B cells. And then outside the light zone, we have the dark zone, which is also swarming B cells, but just in a greater concentration. Now, I want you to think of the dark zone, like dark, maybe like the soot from a blast furnace or something. Dark zone like a workshop, right? Or the blast furnaces, right? And this is where B cells will go to work on themselves in a process called affinity maturation. Now, what is this fancy term? What is happening in this germinal center anyways? A B cell will be out of the bone marrow, everything. It has completed the processes that entail it forming a B cell receptor. By default, it will be in the IgM form, technically IgM and D, but for all intents and purposes, in the default IgM form, which has 10 binding sites, which is fine, but the affinity is not very good. It's not very strong binding, which kind of sucks. So you can imagine why the B cell would want to progress further. So you have this B cell, and mind you, this antigen will recognize an antigen. This B cell receptor antibody will recognize an antigen, and the antigen might not even exist because we discussed it's this big, recombination type genetic processes that produce. Really, to some extent, it's random. So that's why we might get T cell receptors or antibodies for antigens that might not exist. But should it exist, it very well might exist stuck to a follicular dendritic cell, because that's like the antigen sequestration point for B cells, right? So how will the B cell, mature but naive, find the antigen in the lymph node where these follicular dendritic cells are residing at these lymphoid follicles? It's going to want to get into the lymph node, and it'll do so through a structure called a high endothelial venule, or an HEV. And the whole point of this is to direct the lymphocytes from the bloodstream into lymphoid tissue. The B cell is going to find its cognate antigen to its B cell receptor on the follicular dendritic cell in the light zone, right? The B cell will activate, and it will take up shop as an IgM producer after going some additional processes, which we'll talk about, or it will go through the processes that we're about to talk about and then isotype switch, which means it will change from its default IgM producing form into an IgG, A, or E producing form. And we discussed all those previously. So the processes at hand is typically going to involve something called affinity maturation. And this is like a ping-ponging, if you will. Ping-ponging, right? The B cell will spend its time, and really it's quite Darwinian if you think about it, and I'll explain why, in this microevolutionary process. B cell will go to the follicular dendritic cell. It'll snoop around for its antigen. It'll find that, like, aha, I can grab this. But then it will ask itself a fundamental question: how strongly can I bind this? And chances are it will answer, not strongly enough. So it will run off to the dark zone from the light zone where the follicular dendritic cell is, and it will want to undergo some tweaking processes. This is going to be directed by the cytokine environment in the dark zone that really facilitates this type of process to occur. It'll undergo the somatic hypermutation. And basically, this is no fundamental change to its receptor. No, if it recognized receptor A, if it recognized antigen A, it will stay recognizing antigen A. But the extent to which it does so is what changes, and hopefully it will be improved. So then it will go back to the follicular dendritic cell who had the antigen, and it'll be like, okay, I can grab this antigen, but again, it will ask itself the fundamental question: how strongly can I bind this? And if the answer is very strong, the B cell will be quite satisfied with itself, and it can go off to the dark zone over to the B cell T cell interface area, which we'll talk about in a minute, and it will proceed with the maturation process, having the ability to strongly bind an antigen. Now, how is this Darwinian? It sounds like a success story. Well, what if, we have to be a little bit, pessimistic in this case. What if its genetic process in the somatic hypermutation, by the way, which is mediated by an enzyme called AID, not too important right now. What if the somatic hypermutation doesn't make it a strong grabber of antigen? What happens? Well, another B cell will come along who is a stronger binder of antigen, and they will take the antigen for themselves. And if the B cell is without an antigen, it has nothing to show for, it will get killed by something called a tingible body macrophage. They kind of patrol the germinal center, looking out for the weak ones and eliminating them. So really, it's a survival of the fittest. And we should by no means model society by that standard. Some people would disagree, but we're people, not B cells, thank God. but that's kind of where the economy is heading now, right? Anyhow, but that's really what's happening. By this process happening with B cells, you're basically ensuring that only the best B cells are surviving, and by best B cells we mean the ones that have receptors that can bind antigen quite strongly. And that's important in an immune system response, because these B cells who survive will basically undergo the maturation into like memory cells and plasma cells, and these will form progenies. And you want the progenies formed to also have that in their genetics, which is strong ability to bind an antigen, right? So that's why it's called affinity maturation and clonal selection. The ones with good affinity will mature, and those are going to be the ones that will produce a progeny. And clonal selection, because the ones that are weak are killed off by tingible body macrophages. So that's why it's sometimes called a Darwinian or microevolutionary process. Quite remarkable. So now we'll do a profile of each type of secondary lymphoid tissue. Now understanding the lymphoid follicle should make it much easier. In this video, we will just focus on the lymph node, and then in the next video, we will talk about all these pretentious things, as well as the spleen, our blood filter. So what is a lymph node? It is an encapsulated structure. We have many of them, all dispersed throughout our body, and the basis of them being dispersed throughout the body is just basis of regional anatomy. If you have an infection in your foot, you kind of want a lymph node closer to your foot, providing the troops. It would take too long for something from, say, the armpit to make its way down there. That's just regionality. And they are visible in the gross view. You can even feel them. If you go up into your axillary fold, and you can find like these little marble-like things. That's them, right? And they roll around a bit. If they're big, that might indicate some kind of infection, and God forbid, cancer. But more often than not, infection or just some kind of reactive change. So that's important also, because you can appreciate grossly, right? That which is just the macro view, that when there's something like an infectious process going on, and there's like this big proliferation of our lymphocyte counts, you can feel that, or you can see that, I guess, on the basis of a lymph node swelling, right? There is such thing as abnormal swelling, like Yersinia pestis, which is the bacteria that causes the bubonic plague. That one forms something called buboes, and those are just basically like a gigantic lymph node. It's quite atrocious, but that's not normal, so we're not going to talk about it today. But yeah, and also, if you're interested in pathology, you're ever grossing like a specimen, usually like intestine, what has some mesentery and fat attached to it. Usually in the resection, when you're doing the, portioning it up, sectioning it off and everything, you need to take out all the lymph nodes, and they need to be stand interpreted. So digging for lymph nodes sometimes can be a bit of a pain when you're grossing an intestinal sample. But the lymph nodes are grossly visible, so you can find them literally with your tweezers and scissors. But the thing is, if you have a patient that's undergoing like some kind of chemotherapeutic regime or radiotherapy or something of that nature, lymph nodes actually might be quite small, and they'll be very hard to see on the macro view. This means nothing for you unless you're like interested in that field. But I just thought that was interesting. So it's kind of one of those things where you can actually see something like a microscopic change isn't really microscopic. Like you can also see it on the macro view. I mean, that's the basis of anything, because everything kind of happens at the cellular level to some extent, right? And it just has like a larger manifestation, like a domino effect of life, I suppose. Anyways. So I'm not going to harp on this slide because I'm going to basically talk it out as I draw it. So if you prefer to like take notes off the slide, there it is. I'll pause, and then we'll just work through on the next slide. It's here, just same thing, just smaller. Okay. So lymph node. It's going to have these afferent vessels. Afferent with an A just means it's going towards, right? And then efferent with an E is like E for exit, and is going away from. But these are afferent vessels, so they're going to bring lymph, which is a fluid, into the lymph node. And now the lymphatic vessels are throughout the body. They kind of flank the venous system as well. And they are basically ushered along by our muscular contractions, and also like the movement of our diaphragm kind of massages the lymph to move around a bit. So lymphatic tissue is in circulation. It's quite important, and it will hit these little nodes, these little lymph nodes, and that's kind of where the fluid percolates throughout the body. So you have the afferent vessels, they're going to lead into the marginal sinus. It's kind of the first part of the lymph node where they arrive. Not much happens here, but there are some macrophages here that kind of act as a guard, right? So we arrive in the marginal sinus. And the real bread and butter now is going to be entering something called the cortex, right? So the cortex, or the cortices, because there's multiple, is going to be your B cell area. And when we talk about lymph node microanatomy being testable, this is the most testable stuff. Like what and where are things? So what is the B cell area? Cortex. So the way you can think about it maybe is BC, like the time stamp categorization, right? BC, AD, whatever, right? And then the other way, and then there's a more inwards, you have the paracortex. The way I want you to remember that is maybe PT, like physical therapist, paracortex T cell area, right? Anyhow, so this is the B cell area. So if we said that the remember the library for the B cells is the follicular dendritic cell. So if I were to ask you in a lymph node, where might you find follicular dendritic cells? What would your answer be? Marginal sinus, cortex, paracortex, or medullary sinus? I hope you're saying cortex because that's the B cell area, because B cells are swarming these follicular dendritic cells, right? Then more inwards, you have your paracortex. And remember, cortex doesn't mean core. It's cortex like more outside. Medullary tends to mean center. But between the medullary area and the cortex is the paracortex. And the paracortex is going to be your T cell area. And between the cortex and your paracortex, you actually have an interface zone where B cells and T cells kind of flirt and intermingle. It's quite a dynamic setting. Anyhow. So more inwards, paracortex T cell area. And so who presents antigens to the T cells? I hope you're saying dendritic cell. Remember, dendritic cell comes out from the periphery, gulps up its antigen environment upon its toll-like receptor picking up a red flag, slurps up its environment, and then swims to the nearest lymph node, takes about a day, sets up shop in the paracortex. I mentioned that last time, but perhaps last time we didn't know what paracortex was. Now we know. So sets up shop in the paracortex, which is the T cell area, because T cells are going to basically snoop around the dendritic cell for antigen presentation, right? Okay, so we have cortex, paracortex, and the interface zone between them, and then we have the medullary area, and then from the medullary area, basically there's the efferent lymphatic vessels, and that's basically the exit point of lymph from there, because it just percolates through. There's like trabeculation that kind of go through, so lymph will flow through, hit the cortex, hit the paracortex, hit the medullary sinus area, and then flow out. That's also important to know. But this area where the efferent vessel is called the hilum. Also at the hilum is the entry point of an artery, and then there's the exit point, and it's going to form a capillary. And then also at the hilum is the exit point of the vein, because these do receive a blood supply. The only other thing I'm going to mention is something called HEVs, the high endothelial venules that I spoke about last time. Those are the entry points, remember, for lymphocytes as well. And those actually flank the paracortex. This is not intuitive, it's just something you need to know. But HEVs are near the paracortex. And so that's something I've actually seen asked on exams, is kind of like, where is the HEV? And it's near the paracortex. And that's just something to know. And if you kind of vibe with the fact that perhaps paracortex is maybe close to the T cell area and the B cell T cell interface, maybe it's kind of like that's the way it wants to do it. Whatever works for you. For me, it wasn't too intuitive, but HEV, they're important. There's actually believed to be because how HEV really deals with like lymphocyte trafficking. It's believed that some cancers can induce HEV formation, and that can be catastrophic for tumor spread. So it's a hot topic of research. So that's a lymph node. And all right, so we mentioned that we have a B cell area and a T cell area, cortex and paracortex. How do the B cells and the T cells know to localize to these areas left to their own devices? If they just flow into the lymph node, won't they just flood where they flood and it'll just become mixed up and it'll be like a big mass of mixed B cell T cells? How do they know to aggregate in areas? That's because follicular dendritic cell, which is trying to attract the B cell scholars to its library, has a chemokine called CXCL13. It's also called BCA1, which means B cell attracting chemokine 1. I much prefer that name because it's intuitive and tells you what it does. CXCL13 tells us nothing. But guess which one textbooks like to use more? CXCL13, right? So CXCL13 is the cytokine that attracts the B cells, but what's the receptor for CXCL13? How do the B cells smell it? It's through a receptor surface marker called CXCR5. So CXCR5 on the B cell will smell CXCL13. So if something has CXCR5, it will head towards the direction of the follicular dendritic cell in the B cell area in the cortex. And so I'm going to mention this because it actually has some applicability. We have, this is just a rough diagram. So we have outside on the cortex, and in the inside we have the paracortex. We established that the cortex is the B cell area, right? And that the paracortex is the T cell area. Now, we know that B cells are going to localize to the B cell area on the basis of them having CXCR5, so they can pick up CXCL13. It's a very CXCL13 rich area. All right, so I'm just going to draw a little 13 in here. All right. So the B cells will aggregate towards the B cell area. Now I want you to appreciate that between the B cell and the T cell area, we appreciate things in life are a spectrum. There is an interface zone, right? So this is an area where kind of there are both T cells and B cells, just kind of like transition point, right? Kind of like if we're transitioning from red to blue, you kind of expect like a purple zone in the middle. This is the purple zone, if you will. Follicular helper T cells are one type of CD4 cell that is actually famous for having CXCR5. So if it has CXCR5, what might you be able to predict about its activity? You might be able to predict, oh, if it has CXCR5, that means it likes to smell CXCL13 from the follicular dendritic cell. So that you might deduce that will mean that in the T cell area, it probably exists at the periphery in the B cell T cell border, like transition area, just because that part of the T cell area is closest to the B cell area, where follicular dendritic cells smell of CXCL13 is probably more potent. And that's exactly right. You'll notice that follicular helper T cells, which we'll talk about soon, they do localize towards the T cell B cell border because that part is closer, quote unquote, to the heart of the smell, which is the follicular dendritic cell, right? But it's also observed that at some point the B cells will shut off their CXCR5 and upregulate something called CCR7. So you can think of the B cell having two moods. It'll be in the CXCR5 mood, and that will make it want to go toward the follicular dendritic cell. And it also has something called the CCR7 mood, just the other receptor it puts up. This actually attracts it towards the T cell area. So what do you think that does? Well, it's quite romantic, really, to envision that that means there is a subset of T cells who are rushing towards the B cell T cell border, and you have a population of B cells who are also rushing toward the T cell B cell border. Now, though B cells heading towards the B cell T cell border are going to have CCR7, and the T cells rushing towards the B cell T cell border are going to have CXCR5. And again, CXCR5 is the initial receptor that the B cell puts up. But among T cells, follicular helper T cells in specific are famous for having it. Other T cells don't really have it. And that's just because follicular helper T cells are the T cells that want to meet B cells at the B cell T cell border. Okay? So if you can appreciate this, it'll make your understanding much easier down the line when we talk about follicular helper T cells basically being the life partner and the big supporter of the B cell. And that's what we're going to dive into next. So Why do the follicular helper T cells and the B cells want to rush towards the B cell T cell border and meet each other? That's because they need each other. These are life partners. This is a two-way love story because love has to be both ways. Otherwise, it ends in misery, right? How does this happen? The B cell is going to complete its affinity maturation in this Darwinian bloodbath. So not only will it'll pick up the antigen, it'll pick up the antigen strongly. And it will pick up the antigen so strongly, the B cell will become very impassioned, be like, I can hold this antigen so strongly to the point I'm going to take it up and project it on myself like a trophy. And because this is technically antigen presentation, where might it do so? MHC class 2. Okay, so if it's going to be on MHC class 2 and we established that the B cell is going to want to interact with a follicular helper T cell, follicular helper T cell, do you suspect it's going to be CD4 or CD8? I mean, it's in the name. It's going to be CD4 because CD4s are helpers, right? Follicular helper T cell. But still, you can appreciate follicular helper T cell is on the CD4 lineage because it's going to be MHC2 presentation on the B cell who not only strongly picks up the antigen but is now presenting it. So now the B cell, proud of itself, has the trophy projected on MHC class 2. It will put up CCR7 to go to the B cell T cell region and show off its trophy to a CD4 cell. At this point, the CD4 cell is not a follicular helper yet. It has to get a very specific maturation signal to do so. But I'll talk about it now because we'll save some time later. And I know I tend to repeat myself. The really big intention of me repeating myself is that it makes the story consolidate. So that way, hopefully, that by me always repeating that, you can just kind of passively put together the story. And really, all the videos are kind of cumulative in a sense, because this kind of builds on itself. I hope that works for you. That's kind of the style I try to operate here. If it doesn't work for you, I hope you find something that works for you. But I think you will, because you've come this far on your own already, right? And that's not nothing. Anyways. So the B cell and the T cell are now at the T cell B cell border. The B cell is holding up an antigen on MHC class 2. The T cell that has a receptor that also recognizes the antigen will be able to interact with the antigen projected on the B cell's MHC class 2, which is amazing. For me, it's amazing because T cell receptors and B cell receptors, they develop through two distinct processes that have very little to do with each other. And in fact, it's technically a random process because, again, we have T cell receptors and B cell receptors to antigens that might not even exist. But here in this case, they do exist, and they meet. And for me, it's also remarkable because, yes, the B cell and the T cell are recognizing the same antigen. But imagine, well, you'd say if they're recognizing the same antigen, why don't B cell receptors and T cell receptors compete? Well, that would render the whole immune response kind of useless, right? If we have B cells and T cells not engaging in a romance, but rather competing in this toxic type thing. That's not the case, though, because yes, they bind the same antigen, but they bind it on different epitopes, which I think is remarkable. An epitope just refers to like one protein region on the antigen. But same antigen they recognize, but on two different points in a way they don't compete with each other. For me, this is completely amazing because two different processes yielded the recognition of the same antigen. But despite that, even they know to take it one step further and recognize the antigen at different parts so they're not competing. It's just phenomenal. This is probably one of the most amazing things I've heard of. My passions aside. So, what do the B cell and the T cell do for each other? Well, it is a romance, so they're going to passionately interlock. But they're a little bit better than people because they can do so like 6 different ways. And so what's going to interlock, right? The B cell antigen on MHC class 2 is going to be presented to the T cell receptor, right? So that's one binding point. And then you got a couple other binding points because there's like the co-receptors and all these things that I didn't really talk about too much here, but those are also part of it as well. So you can add those to the count. What else? The B cell has CD40, and that's going to bind T cell CD40L. And we've spoke about this before. This is a co-stimulatory signal. Okay, so that's happening. You also have the B7 on the B cell connecting to CD28 on the T cell. And remember, in this capacity right now, the B cell is operating as an antigen presenting cell, and that's where B7 exists. So before we really spoke about B7 in the context of, say, a macrophage or a dendritic cell. But mind you, B cells are also antigen presenting cells. While they're not necessarily phagocytic, they are antigen presenting cells. So they do have B7. And remember, B7 has the other name CD80, 86, because B7-1 is 80, and then B7-2 is 86. I just call it B7, but just know it's the same thing. And that binds to CD28 on the T cell, right? And then another thing that we didn't talk about, because this is kind of more specific to this region in the body, is something called inducible co-stimulatory, I think it's like factor or molecule, ICOS. It is a surface marker on the T cell, and I remember it's on the T cell because T ICOS sounds like tacos, Ticos tacos, right? And that's going to bind ICOS ligand, right? So ICOS ligand is on the taco, on the T cell, ICOS, right? This is a co-stimulatory marker needed for maturation of the CD4 cell into specifically a follicular helper T cell. That's part of the signal needed. Now, just like anything in order to happen, it's multi-step. So at this point, you can really regard the CD4 as a pre-follicular helper cell, right? And the basis of that is that anytime you have the maturation of the CD4 cell into a different lineage, Th1, Th2, Th17, etc., that really happens because of the cytokine environment. We kind of spoke about the dossier before with the dendritic cells and everything of that nature. The way it's happening now is that you have the CD4 cell. It will get signals that will induce it in that direction, but it's still being studied on what induces a pre-follicular helper T cell to form. And this is not testable whatsoever. Right now, some researchers finding, based on some mouse models, that interleukin-6 can induce follicular helper T cell formation, which is interesting for me because interleukin-6 is a pyrogen, much like interleukin-1, and they just kind of guide like that heat response in the immune system. But it's found that it can actually induce follicular helper differentiation. And so once it's kind of at that if you will, a follicular helper T cell stage, what will seal the deal to make it a full formal follicular helper T cell is this connection of B7 to CD28, antigen to MHC class 2, ICOS L to ICOS. Now you have a formal follicular helper T cell produced from this mutual maturation type process. And what happens to the B cell? Well, it becomes fully mature. It already came into this whole love affair with a strong antigen. I mean, this strong antigen binding capacity because of the hypermutation. Mind you, this whole thing only happened because the B cell is able to strongly bind and then present the antigen. So it's kind of like en route to wanting to isotype switch. It becomes hypermutated, becomes good. So, but that's the end goal here. In the fully mature form, now the B cell can isotype switch out of IgM into IgA, IgD, IgE, away from the default IgM, and it can enter the lineages now of becoming a memory cell or a plasma cell, which basically means it's an antibody factory. And it can do so staying in the lymph node, or it can go out into the periphery, closer to the infection site, and crank out antibody there. As opposed to the follicular helper T cell, it's actually going to take up shop in the lymph node and it will maintain the environment there. And what do we mean by that? It will basically produce cytokines and give survival signals that will allow long-lived cells to be long-lived. So they are long-lived because the follicular helper T cell will maintain an environment that facilitates that, right? It's not as hostile as the rest of the body. And it's actually interesting to know that people who have deficient ICOS actually have a deficient adaptive immune system, and that's simply because the concept of this mutual maturation can't really occur too well. Therefore, you really don't get quality B cells, And when you consider that IgA is the most common antibody in the body, you can imagine why the process in which you produce IgA being impaired can be quite problematic, right? So that's what that is. I know this we spoke a lot on the slide, but this is the bulk of it, really. This is the bread and butter of today's presentation: mutual maturation T cell B cell border. I mean, when we speak about B cells becoming antibody factories, this is how it does it. Welcome to the lymph node. You can see why they're quite important. Why they're quite important. So this is it put in different terms. I invite you if you want to take a look at this, but I just basically go over the schema again. I mean, we can go very briefly from the beginning. B cell is going to enter via the high endothelial venules. It will go over to the cortex B cell area. It'll go to follicular dendritic cell in the light zone. It will find its antigen, and it will try to capture that antigen and present it. But in order to do so, it has to get better at capturing antigen. So it will ping pong between the light and the dark zone, undergoing somatic hypermutation until it can pick up the antigen strong enough to a point it can take it up and present it. And I'm not sure if I mentioned this, but it's actually quite funny. There's some research to suggest that the B cells, when they're like doing this antigen testing back and forth, they can actually rip off antigen from other B cells, right? And that kind of what makes it Darwinian as well. Like they can literally rip off the antigen from another B cell, and the tingible body macrophage would be like, you're a loser. You got your antigen taken away, you must get killed. And so it's very Darwinian. So tingible body macrophages kind of patrol the follicle, will clean up the weak links. And again, the whole point of having this process to ensure that we're only getting cream of the crop antibodies later on, right? So anyways, once the B cell is able to effectively hold its own and grab an antigen and do so quite well, and then be able to present it on MHC class 2, it'll go to the B cell T cell interface zone, meet a follicular helper T cell. That T cell and it will interlock passionately, very passionately, and undergo a mutual maturation. And the B cell will become its mature form. It will isotype switch, should it need to, into an IgG producer, and it will set up shop in the lymph node, becoming an antibody factory, or go out to the periphery, hence the left plasma cell, or it will commit to becoming a memory cell, which basically means that should the infection come along again in the future, you don't have to go through this whole process again. You can just spit out prepared B cells from those memory plasma type cells, right? Memory B cells, we call them. And then as for the T cell, it'll be able to undergo its full maturation from a CD4 to a kind of a pre-follicular helper T cell into a full-on follicular helper T cell, which will basically maintain the lymph node, allowing long-lived cells to live for a long time and produce the cytokine environment that allows the dark zone to kind of be a workshop area for B cells trying to hypermutate. It just basically maintains the follicle. And it's a beautiful story, the B cell and the T cell. Especially if the B cell stays in the lymph node, they're kind of paired for life, kind of like taking care of each other and taking care of you. It's a beautiful romance. Of all the romances that exist in physiology, I think the follicular helper T cell and B cell are by far my favorite. But that's that. So I think I mentioned all these housekeeping notes along the way. Last thing I think I maybe want to bring up, it's more so a histology term. If you hear the term centroblast and centrocyte, that just refers to the B cell ping-ponging between the light zone and dark zone. So when it's very immature, and bigger, it's going to be called a centroblast. And then when it's smaller and more mature, so it's closer towards entering its last round of somatic hypermutation and all these things, and it's quite ready to almost go off to the B cell T cell interface, we call it a centrocyte. And it's smaller, and so blasts comes before cytes. It's like anything, it's like blastic. It's like earlier phase, and then it matures, and so the blast is bigger, right? So B before C, B for big, right? So if again, I don't think you'll hear that term too much, but if you hear it, this lecture prepared you, or maybe it did. I hope it did. That's the whole intention of these, right? And again, those B cells who lose the competition will get killed off by tingible body macrophages. And again, it's quite funny, but sadistic also to think that B cells can rip antigens off each other in competition. Again, not a way to run a society, but it is a way to run a lymph node because, again, we get quality antibody, hopefully, right? So, last thing, I will ask you, are your ducks in a row? In respective order, please tell me where each of the following, what each of the following areas are associated with. And so I'll pause the video now and smile so you can think it out. Unpause. All right, let's take it one by one. The B cell area, that is the cortex, okay, BC, okay, and that's more outer, right? And then we work inwards towards the paracortex, which is the T cell area. So those are the first two answered for you. Dendritic cells, well, if you want to think, don't just rote memorize. What is dendritic cell associated with? This is like lecture 5 or 6, I believe, that we spoke about this more. Well, dendritic cells do the antigen presentation to the T cells, right? And so that's going to be like paracortex association because it's paracortex is the T cell area. So dendritic cells will be associated with paracortex. How about follicular dendritic cells? They're the great library of Baghdad for the scholar B cells. And so if it's the B cell area, then we're thinking cortex. Very good. So cortex, paracortex, paracortex, cortex, because follicular dendritic cells are in the B cell area cortex. And then HEVs, high endothelial venules, I suggest to memorize where those are, and that's paracortex. That's where they're near, right? So the answer would be B, right? So again, when we talk about the microanatomy for testable purposes, just knowing what is where, and then being able to deduce from that what processes are occurring in whatever region. If you can kind of make a story out of it, like understand what's happening in each part, you'll be in the gold. And I know today we really spoke mainly about the cortex. But in the past video, we really spoke about like the CD8 maturation and everything, because that's the T cell area. We've already discussed paracortex in great depth, but now we have a little bit more appreciation on where it is in the body, right? So again, paracortex, T cell area, cortex, B cell area, and I think you'll be golden. I think you'll be quite adept going forward talking about lymph nodes. So these are my sources. I hope this was helpful for you, and I thank you for your time and attention. And I hope to see you in the next one. All right, everyone. Have a good night. Take care.